let's talk about our innate immunology. And again, a lot of times these are referred to as non-specific defenses. What that means is these defenses work for pretty much any exposure to any organism. So any microbe or any virus, these will be the defense systems that will first kick in. So we'll talk about the difference between innate and adaptive, and we'll talk about how these lines of defenses work. So first of all, we've been talking about disease and microorganisms this whole semester, and now we're going to be talking about ways that we can keep ourselves safe. And so the idea of immunity is it's anything that allows us to be able to protect ourselves against microbes. So we've been talking about how some people were naturally resistant, for example, to COVID. It's because they had a unique immunity in their bodies. And we talked about how our genetics can be different and affect our immunity. Some people, unfortunately, will get sick very easily. So they might have a different susceptibility, meaning that they have a lack of immunity. So their immune system doesn't protect them too well. We have two general mechanisms in place to protect us. We have our ones that we're going to talk about today, which are innate. We also, as I said, call these non-specific because they target everything the same. And then we also can have adaptive immunity, and those are going to be our antibodies. Those are ones that are learned and specific for each organism that we come into contact with. So, for example, when you get a vaccination, you are building antibodies that are specific to that organism. So I've had a couple people ask me, well, why do I have to get a COVID and a flu shot? They're both viruses. Why doesn't one cover the other? Well, exactly. They're different families. They're different structures. They're entirely different viruses, right? So that's like saying, well, I'm going to get a dog, and I bought a dog crate that's this big. That should work, right? And then you come home with a German Shepherd. Is that going to work if you have a little? Yeah, it's not going to work, right? So not just because it's in a general category doesn't mean that they're the same. So that's one thing that's going to be different with our adaptives. Now, when it comes to our host defenses, the first lines of defense are the ones that are going to try to keep organisms out. So these are going to be things like our skin that's in place to try to keep organisms away from us. This is going to be things like our mucous membranes, like our eyes, nasal secretion. If something does get inside, though, we do have a second line of defense. These are going to be our white blood cells. Our white blood cells are going to be a general line of defense. It's going to target any invader the same way. We also have inflammation and fever that can help protect us, and we'll talk about how that works. And then, if those first two don't help us, we get to our third line of defense, and that's the adaptive immunity. Those are the antibodies. So our body has a whole bunch of different defense barriers in place. And the idea is that we try to make it harder and harder and harder for the microbe to actually cause illness. Now, innate immunity is one that's always present. It's not specific, so it attacks everything the same. It acts against all organisms the same way. And it's an early prevention system. If we go to adaptive immunity, which we'll talk about next time, again, that's going to be specific. It tends to be slower the first exposure time, or the first time it sees a microbe. This is why vaccinations are important. The first time you get exposed to something, your adaptive immunity has to learn how to fight it. And that's what the vaccination is. It's a learning time. And it's also a recognition time. I told you it's like a most wanted poster. That way, when you get exposed the next time, you have what are called memory cells present. A memory component means it's an antibody that will recognize what you've been exposed to and target it very quickly. So we have to have a slower response first to get this fast response. And it's going to involve special cells referred to as lymphocytes that are also known as antibodies. So if we were to kind of pictorially show this, this would basically show our first line of defense, again, is our skin, as our mucous membranes. Our second line of defense are our general white cells, and then that third line of defense are our antibodies. So which defense system, innate or adaptive, is going to prevent the entry of microbes into the body? Adaptive. Okay, so what does adaptive immunity cover? Antibodies. Do antibodies prevent the entrance? No. So it would be innate. Okay, so innate, like your skin, that's preventing the entrance of a microbe. Mucous membranes, things in our nose and our lungs, are preventing the entrance. So that would be innate. Overall, our body has how many lines of defense? 3. Excellent. We have 3 lines of defense. So the first 2 are going to be our innate, and our third one is our adaptive. All right. So let's talk a little bit more about those first line of defense mechanisms that we have in place to keep us safe and healthy. The first one is going to be our skin. And our skin is very important to protect us and keep microbes away. The top layer of our skin is completely dead. So if you look at your skin cells, the top epidermis layer are composed of dead cells. We shed the top of our skin constantly. When you see dust in your house, that's made of skin particles. And it's kind of like gross to think about, but we constantly shed. The idea then is if a microbe lands here, there's not a lot of nutrients, there's not a lot of moisture. It's kind of like the microbe landing in the Mojave Desert. It's not a great place for it to grow. Not only that, if it tries to dig in and kind of burrow into a skin cell, we're going to shed that cell later. So it's not hopefully going to get in too deep. That's the idea, is that this layer is just constantly shifting like sand. We also have to have our skin intact so that there's no space in between the skin cells. If we have a space, if we have any kind of a canyon, we're giving them an entrance. This is why if you get a cut, that's why it's more likely to get infected than the skin around it. That cut is an entrance wound. So microbes are going to look for any kind of way that they can breach this defense system. Our skin tends to naturally be very dry. And again, that dryness is kind of supposed to be like a desert region. It's supposed to be arid. And the idea behind it is that if it's arid, microbes aren't going to be able to be intact. It's going to be harder for them to live. Now, areas of the body that are naturally moister, like places where we sweat, or if you have skin folds, that's where you can end up having more of a common occurrence with skin infections. So for example, under the breasts, under the armpits, in the genital region, in the feet, those areas tend to be moister, and they tend to have more of a chance of getting like a fungal infection. So yeast, fungus, anything that's able to survive a moisture environment will start to grow there. This is why a lot of times patients that are obese also have problems with skin folds. They have to constantly put powder to keep their skin dry. Now, how does the skin work? Well, basically it works because it has a special protein in it called keratin. And keratin is something that will accumulate as the cell starts to mature. And what happens is the keratin is basically going to completely take over the cell. The cells in our skin start to produce a whole bunch of keratin. And the keratin is like a waxy component that is waterproof. Now, sadly for the skin cell, when it produces this waxy waterproof protein, it works to make the cell impenetrable. It's completely waterproof now. But it also makes it so that the cell can no longer accept nutrients from the outside. And the cell can also no longer push waste products outside. It basically seals it up like a tomb. This is why our cells end up dying. So they do this as a sacrifice for us. I mean, isn't that sweet of them? I mean, they end up producing so much keratin that as they produce more and more keratin, that keratin builds up and it actually causes them to die slowly of toxins that are inside of them that they can no longer get rid of because of that boundary layer. And so eventually, by the time they reach the surface of our epidermis, they are completely dead. They've lost the ability to survive. I mean, that's a huge sacrifice. Now, if we look at the dermal tissue, which is the layer of skin where they're first starting to grow, what we can see is that those cells are living. As they start to move up towards the epidermis, we get natural cell migration towards the surface. This is where they start to produce more and more keratin, and this is where they start to become impenetrable and they start to die. So that's why the outer surface of your skin is dead, the inner surface is going to be living. Mucous membranes. So mucous membranes are important for inhibiting microbes. And the idea behind this is the mucous membranes are trying to wash the microbes away before they can gain access. So, for example, we have mucus that we naturally produce in our noses, in our lungs, and the idea here is that microbes will stick in this mucus, and then we can cough to get rid of them, or we can blow our nose and that will get rid of a mass number of microbes that have been collected. Now, we do run into trouble sometimes because there are a few pathogens that like mucus. They've adapted. And so if our body's producing excess mucus, they start to grow. They don't care. These are the ones that a lot of times will cause those secondary infections. Remember we talked about how after we have our period of illness, we can get a secondary infection. That's usually from organisms that can grow in mucus. So if we look at mucus, for example, we can have our goblet cells that are involved in producing enzymes that are going to be released in, for example, the nose and in the lungs. And the idea behind this is the mucus is sticky. It'll trap any microbes that we breathe in or that we come into contact with in the cells. And then that mucus, again, can be moved away from the body. A lot of times the cells will have cilia attached to them. We've talked about cilia. They're like little oars that will kind of beat and move away from the body. So if I have mucus here in my lungs, it'll capture the bacteria and then the cilia will push it back up, which will make me cough, and then that will get rid of the microbes. If I have cilia in my nose, it'll capture and push the microbes back down, so I sneeze. That'll get rid of the microbes. Another physical factor I can have involves the lacrimal apparatus. This is our system that involves blinking or creation of tears. So whenever I have something get in my eye will start to water. If it's windy, my eyes can water. The idea behind this is that our eyes naturally produce moisture that we can blink and wash away microbes. It's kind of like when you wash your windshield when you're driving, that's pretty much what you're doing. Your eyes are constantly producing solution that we blink to get rid of our microbes. Now, this is why we blink. And have you ever noticed you don't think about blinking until somebody mentions it? Like when somebody says, how many times do you blink? All of a sudden you're like, and you start thinking about it. It's like this weird thing. But yeah, the reason we blink is it's kind of like windshield wipers. We're constantly washing our eyes to get rid of any microbes that have come in contact with us. Other physical factors that cleanse kind of like tears would be things like saliva. Saliva keep our teeth clean. They try to keep harmful microbes out of the mouth. Urine, whenever you go to the bathroom, you're flushing out the urethra and the bladder. So the idea is you're trying to get rid of bacteria. And then vaginal secretions. Vaginal secretions have a bad rap. A lot of times people try to change them. There's a process called douching where you try to get rid of vaginal secretions. What you're doing is you're actually affecting your natural immunity. You're changing the pH of the vaginal region. The vaginal region should naturally be acidic. And what do we know about bacteria with acid? Do they like that? No. And so the idea is it's a natural defense mechanism. We see a lot of people who use douching or flushing in the vaginal region that are prone to vaginal infections because they've made their pH more neutral and then the bacteria like it, so they'll start growing. So these are all natural things that are going to get rid of our bacteria. Here's some bacteria growing. Aren't they cute when they color them? I mean, I just kind of wish they looked like this in real life, right? They don't really look this bright and colorful. But you can see we have like different varieties of bacteria growing in the tissues here. Another first line of defense are the hairs in our nose. This picture just makes my eyes water. Waxing your nose hair. Oh, I can't even imagine it. It hurts so bad. So, should you wax your nose hair? Is that a good idea? No. Excellent. Good job. No, it's not a good idea. There is no microbiologist that will go and get their nose hair waxed. I'll tell you that right now. And the reason for that is because if you remove the nose hair, you're removing a defense. Now when you breathe in, you don't have these little curtains, and that's basically what they're acting like, are little curtains that are going to trap the microbes. So when they're gone, microbes have a faster route to get into your face and into your nasal cavity. Not a good idea. We don't want that. So definitely recommend not, going in and getting your nose hairs waxed. Another example of a physical factor would be a ciliary escalator. That's where I mentioned the cilia kind of move together to move microbes out. So that's called the ciliary escalator. They trap and move microbes away. Now remember, some microbes can affect our cilia. Here was one we talked about, Bordetella pertussis. Do you guys remember what it causes? Whooping cough. Excellent. And remember, the reason whooping cough is so damaging is it would cause an excess of mucus that would cause patients to basically no longer be able to breathe. They would drown in their mucus. The reason it works that way is because, remember, they cause paralysis of the cilia. So we need to have those cilia working. We need to have them being able to push microbes out. Because if they don't, the microbes just build up inside. And that's exactly what happens with whooping cough. We get this big congestion block in our lungs. Another one is defecation and vomiting. I know nobody really wants to talk about this, but if you're sick, it's a great way for your body to expel microbes. It's like hitting the eject button. There was a study that was done, this is called Vomiting Larry. It was a robot, and then they would put different types of food, different sized particles in Vomiting Larry, push a button, and they would film against the backdrop Larry projectile vomiting. The idea was to try to show how far small particles could spread. And when they filled Larry up with viral-sized particles, they found that when you projectile vomit, the particles can spread over 10 feet. What this means is when you're in the healthcare setting, if you have a patient who's vomiting, make sure that you have a mask on. You want to protect yourself as best possible. Because of the fact that if you're in a 10-foot radius, you're getting exposed to whatever they have. So make sure you're always protecting yourself. Wouldn't that be a fun job to do, right? What did you do today? Oh, I worked with Vomiting Larry. It's like, okay, there you go. Yeah, so different projectiles would travel different distances. And I put the link there in case you ever want to read about them. Okay, so who can tell me one physical factor that we just talked about and how it works? Yeah. Lacrimal apparatus, excellent. And what does that produce? Tears, good. And those tears are used like windshield washer fluid. They basically wash away the microbes, right? It turns out that our tears, and we'll talk about these when we talk about our chemical defense, they are actually going to be very antimicrobial. So the tears that we actually produce, the actual chemicals in the tears, can kill microbes. We normally do a lab where we gather our tears and we see how they work. With COVID this semester, we're not going to do it. So it's listed, but we're going to cross it off just because having infectious biologicals in the lab during COVID, probably not the best idea. So I was like, I was right at that edge and I was like, okay, so we'll cross that lab off. But the idea is that if you collect tears and you put them on those discs, like what we use for cleaners, and then you make a plate with staph and E. coli, your tears actually inhibit them just like those cleaners did, which is pretty amazing. Usually to create tears, I get you guys fresh onions. And so we cut the onions and smell the onions to get the tears. One year, our normal lab tech was on vacation. The other lab tech was helping out, and she put the onions in the fridge, which isn't good. It gets rid of the enzymes that make us cry. So nobody was crying. So I was trying everything. I was like, I'm going to give you all Fs. And they didn't cry. Even then, I was like, oh, you guys are hardcore. I was trying to think of everything. And I didn't cry.
Yeah, I was going to ask how you were going to make us cry.
I know, because I don't want to give you anything too toxic. The onions are looking really well. So what we found, though, that worked really good, do you know how we all ended up crying that day? We watched the first 5 minutes of Up. Oh, that movie. I know. We were all sitting there. It was like we were trying to find sad movies on our cell phones. It was hilarious. Like all the kids were watching different movies, but we found that Up worked the best. You know, if you've ever watched Up, the first 5 minutes, they just take your heart and throw it on the ground. You know? So that worked. So there you go. If you don't have onions nearby, you can watch Up. Okay, so those were all physical factors. Things that are working to keep microbes away. Now, these work hand in hand with chemical factors. So a lot of our chemical factors will have special enzymes in them, special proteins, and they work with the physical factors. So a chemical factor would be something like sebum. So I mentioned that we have our skin and that our skin is dry. That's the physical factor. Sebum is a secretion of the skin, kind of like our sweat. And what its job is it provides a protective cover over the film of the skin to try to keep it waterproof. It also lowers the pH of the skin. If I was to check your skin right now, you would be acidic. Again, that's good. We don't want to have microbes growing, and acidic conditions make microbes unhappy. Now, sebum is a natural solution that we produce in our skin and in our pores, but some of us produce more than others. And remember, we had our organism that produced acne, and it was living by eating sebum. Does anybody remember the genus that produced acne? Excellent. Propionibacterium. Excellent. Oh, you guys are making me proud. Okay, so Propionibacterium was the one that created acne. It's one that likes to eat sebum. So we do have a few microbes out there that can eat it as a food source. Most of them are inhibited by it though. So when we sweat, when we're hot, those solutions that we're producing are antimicrobial. Okay, so it's not a bad thing to sweat, or as us girls do, we glisten. It's not a bad thing to glisten, all right, because we're producing antimicrobials. I mentioned tears, and I mentioned that our mouth can be protected through saliva. The reason for that is because tears and saliva have special lysozymes in them. Lysozymes are enzymes that rip apart cell walls. So they will break cell walls apart. They will break cells apart naturally. They cause cells to be unstable. So any microbes that come into contact with our tears, our saliva, our perspiration, our sweat, our mucus, because those all contain lysozymes, they are deadly. So they can kill off microbes very quickly for us. So this is a great defense system. Yes, Brown.
You said sweat is antimicrobial?
Yes.
I remember at the beginning of COVID, when the gyms were opening back up, people had to wear masks, and they were blaming it on oh, I'm sweating under the mask, so I'm breaking out.
And that's true. So what happens with your mask on when you're sweating? What can happen if I'm not changing my mask enough? Yeah, I'm getting dirt and debris and bacteria on the mask. And then if I'm not changing it, it's just sitting against my face. And those numbers are building because it's warm, so it's like a little incubator against my face. So as long as you're changing your mask, you're good. But how often do people really change their masks? There was a survey done that was saying that people change their masks once a week.
Oh, that's gross.
Oh, yeah. If you're gonna do that, for example, I have kids. They will not change their masks. I was buying them cute little Old Navy ones, and I would pick them up from school and they're wearing the same one. I was like, wait, you had a different mask on. Well, I like this one better, it's pink, or I like this one better, it has a dinosaur. I finally got rid of all their cute ones, and they now just have ones that get thrown away. Because that way they'll change them. Because I realized they were wearing them 4 days in a row. Kids are disgusting.
Yes.
And I was like, no, they're already Petri dishes. We don't want them wearing masks 4 days in a row, right? That's gross. And so the idea, I just kind of laughed because I had a friend of mine at the beginning of the pandemic, and she's like, yeah, I went ahead and bought another mask, so she had a total of 2. And I'm like, I have 20 right now because I was rotating. I was washing them every day kind of a thing and rotating. So as long as you're changing all the time, you're good. But you're right, if they're trapped against your face, it's kind of gross. Gastric juices. So when you eat food and it goes into your stomach, that pH in our stomach and the gastric juices that are inside of our stomach are going to be important in killing microbes. Now, we did have a few that are able to survive the acidity of our stomach. We talked about one that caused ulcers. What was the name of that guy?
Helicobacter.
Excellent. Helicobacter pylori. Helicobacter pylori. That was the organism that was the exception, right? We said that one liked to live in our stomach. It liked the stomach acid. But for the most part, most of them are going to end up dying if they get consumed. In our stomach acid, also, we are going to be able to neutralize a lot of their toxins. So if we end up eating food that has bacteria in it and bacterial waste, that would be a toxin, most of those would get broken down in our stomachs. Now, sometimes there's such a high toxin level that we can still get sick from our food. That's part of food poisoning. If we eat food that is full of bacteria and full of toxins, then our body's going to hit the eject button and we're going to be throwing up and having diarrhea to get rid of the bacteria and the toxins that will not destroy by our stomach acid. Other pH-related protection, we've already mentioned vaginal secretion, so all of these are going to be important with having a low pH. Another part of our first line of defense that we don't want to ever forget are our normal microbiota. So remember, they are going to keep organisms away, and mostly it's through competition. So in other words, they've already established their homes, their spaces, their nutrients. If somebody tries to come in and take their spot, they're going to be pushing them away. We have daily battles. There are fierce competitions going on right now that we don't even see at the microscopic level, where some microbe is trying to move in on our normal flora and they are fighting it. They're releasing toxins, they're trying to kill off that microbe, and for the most part, they win. And how do we know they win? Well, look at how healthy we are. Think about how very seldom you're sick. Those lines of defense work really well, if you really think about it. So what would be an example of a chemical factor? Who can give me one?
Saliva.
Saliva, sebum, good. And what do they have inside of them that make them good or helpful?
Lysozyme.
Lysozymes, exactly. They have lysozymes that are special enzymes that are breaking apart cell walls. So that's going to be the most important thing in there. So breaking apart the cell walls. Okay, occasionally we have microbes that get past that first line of defense and they go into our second line of defense. Our second line of defense are going to be our white blood cells. So if a microbe breaks through that first line of defense, we now run the risk of having an infection occur. So the second line of defense is called in. This is kind of like the National Guard being called in to help out. They're going to come in and they're going to try to neutralize the threat or get rid of the microbes that have made it past our defense system. So our defensive cells that are going to come into place are our white blood cells. Those are also referred to as our leukocytes. When they're present, they're going to try to actively isolate and destroy whatever microbe has breached our first defense system. They are also going to trigger other responses in the body. So our body is going to start going through a process of inflammation, potentially fever, and then we're going to produce more antimicrobial substances. So this is kind of a chain event that's going to occur. And the idea is to try to wipe out the microbes before they get too established. We're trying to get their numbers down as soon as possible. So if we start looking at the white blood cells, the first thing we notice is that there are different types. If I was to take a blood sample from y'all right now and put it onto a slide and do a blood smear, I would see a whole bunch of red blood cells, but I would also see white blood cells intermixed. And these are pictures from blood cells showing what the white blood cells look like. So these are from blood samples. So the first one at the top are the neutrophils. The neutrophils are considered to be the ones that are first to an infection site. They're the first responders. So they're the first on the scene. If there's a cut or a wound, they're going to be trying to protect us. They live in our blood, but they can quickly move to our tissue. So if I get a cut here, they're going to leave my bloodstream and move into that cut region. And they act as like a little barrier. They start to build up like a little brick wall or defense system, trying to keep the microbes out. They are able to do phagocytosis. Does anybody remember what that means? I mentioned it a long time ago.
Like endocytosis.
Not endocytosis. Endocytosis is pulling in. Eating. Phagocytosis is cell eating. Yeah, so that means that they're going to be consuming or destroying the cells. So you're right, they do use endocytosis to pull them in, but phagocytosis means not only do they pull them in, but they destroy them. So they actually break them apart. The next white blood cell that I would see in a smear is the basophils. Basophils are responsible for releasing histamine. So histamine is responsible for producing inflammation. We run into basophils anytime we have somebody who has allergies. So for example, if you have hay fever or if you have pollen allergies, your body gets confused and it thinks the pollen particles are microbes trying to get inside of you. It just is because your body's a little bit confused about it. So what it does is it sees these pollen molecules or these pieces of hay molecules and it's like, oh my gosh, cause a defense system. We've got something trying to breach in here. And when that happens, basophils will flood to that site and they will start producing histamine. And what histamine does is it causes swelling and inflammation to allow other white blood cells to move to the area. Now, when you have a hay fever attack or a pollen attack, what do you notice happening to your face? Red. Yeah, it gets puffy, it gets red. What about your eyes? They get itchy and they start to water and run. So what happens then is when we have the basophils releasing histamine, it's causing inflammation or redness. It's increasing eye secretion to wash away the microbes. It increases mucus to wash them out of your nasal system. And this is why all of a sudden you can start sneezing, coughing, eyes running, you just turn into a mess. Okay? I mean, it's not, there's no other way to describe it. Believe me, I have allergies to certain things, and it's just like, good Lord, what's going on? It's like, poof, it's not good. It's not a good sign. All right. Eosinophils. Eosinophils are ones that are going to be produced against parasites. So think of things like worms. Think of things like, microbes inside of us. So if I have malaria, if I have worms, I'm going to see an increase in your eosinophil count. So this is kind of helpful for me. If I'm trying to rule things out as a doctor, I will do a white blood cell count, and I'll look to see how your eosinophil level is. If it's super high, that can indicate that you might have the presence of a parasite. So I can use these as kind of a factor to kind of give me a hint as to what's going on inside. Dendritic cells. These are going to be ones that, again, are able to do phagocytosis. But the most important thing about these is that they are going to activate adaptive immunity. So dendritic cells are the ones that are also going to help trigger antibodies later. We'll talk about those when we talk about our antibodies. Monocytes. Monocytes are ones that are, when they're young, they're not phagocytotic. They just live in the bloodstream. They're very small. When they mature, though, they become what are called macrophages. And then they live deep in the tissues. And they stay in the tissue region, and they're going to be extremely phagocytotic. So they move from the blood, when they're young, into the tissue. And now that they're there, they can break down anything. So they're very, very helpful. And they stay in the tissue region, and you can see that they look different when they're mature. And they're ready to attack anything that ends up being able to break through our skin barrier. So very, very defensive. And then we also have our lymphocytes, and these include our natural killer cells. NK cells are really interesting. We're doing a lot of research on them. What we found with NK cells is that they can help identify tumor cells. So for example, when we have a cell that's starting to replicate out of control, a natural killer cell will start to surround that cell and call in other natural killer cells, and they will try to kill the cell that is no longer growing properly. So they'll surround the tumor, and they'll try to start releasing toxins to kill the tumor. So natural killer cells are under a lot of study right now. They unfortunately sometimes can also, though, get triggered by things that they shouldn't. And so there's a lot of research being done with natural killer cells and women who cannot carry babies to term. What they're finding is that sometimes there are women who have high levels of natural killer cells in their uterus, which means that when they get pregnant, the natural killer cell starts to target the developing fetus. And then it will cause a natural miscarriage because it's thinking it's something that's going to cause harm. So there is a lot of research being done right now with this in infertility. What they're finding is if we give women that are pregnant that have infertility issues steroids, the steroids will lower their immune system, lower their natural K cells, and allow the baby to come to term. So it's something that they're doing a lot of research on. Yeah, Rachel? Yes, please do. Thank you. I know they're having a party out there. It's like, I want to go out there and have a party too. Thank you. So with this then, our leukocytes are going to be, again, our white blood cells. And we can tell a lot based on their numbers. So leukocytosis is an increase in white blood cells. If I see an increase in white blood cells, what could that indicate in that patient? If I do a sample and they have a lot of white blood cells, what is that telling me? Yeah, that they have an active infection. Excellent. So if I do a blood sample and I see a whole bunch of white blood cells, that can indicate that something's happening and their immune system is on high alert. So that indicates that they have an infection somewhere in their body. My goal is to figure out where it is. I can also get leukopenia. Leukopenia is the exact opposite. This is where I see a lower than normal count of white blood cells. If I see a lower than normal count, does that mean the person's super healthy? No. It means that the person is unable to produce white blood cells, or something's happening and they're not reaching maturity. So now I need to look at their immune system. Something's going on in their immune system. What is it? Is there a virus affecting them? What's going on? Is there something wrong with the bone marrow? I got to figure this out. So differential white blood cell counts are extremely helpful. They tell me how many white blood cells are there, what kind of white blood cells are there, and they give me a glimpse into that patient's health. If I do a differential white blood cell count, these are the normal counts that I should see. If I have a blood smear in front of me and I count 100 white blood cells in that blood smear, I should see a percentage of about 60 to 70% being neutrophils, about 0.5 to 1% being basophils, 2 to 4% being eosinophils, 3 to 8% being our monocytes, and 20 to 25% being our lymphocytes. Now let's look at these numbers. Do these make sense? Which cells of these are the first responders?
Neutrophils.
The neutrophils. Does it make sense that I would have more neutrophils?
Yes.
Yeah, I want a lot of first responders, right? They're going to be the ones that are going to help me a lot. Which one of these were involved with allergies? Basophils. Yeah, and allergies come and go. They're seasonal, right? So I don't always need those. So it makes sense that they would be the lowest, right? And so then the other ones just kind of average in between. But the idea then is their counts make sense, okay? So this is a normal count. If I see higher or lower than this, then I need to figure out why. I need to resample and I need to try to figure out what's going on with the patient. So white blood cell counts are going to be very important. They're going to give me a big clue or a hint into disease. They're going to let me know that there's different diseases out there. They're going to let me know different things that I might be seeing. They'll tell me whether or not it's parasites or not. If I have an infection occur, I should see differences in which white blood cells are present. So for example, if I have a staphylococcus infection, I should see my neutrophil levels go up. My first responders should go up. If I have a viral infection, I should see my lymphocytes go up. If I have pollen allergies, I should see my basophils go up. So they're going to circulate in our body, and they're always looking to see if there's anything that they need to interact with or attack with. Now, where our white blood cells circulate is through a special system referred to as the lymph system. The lymph system is very similar to our blood system, to our blood vessels. The lymph system kind of parallels it. And basically what happens is the lymph system is kind of this clear fluid that flows beside our blood cells that is full of white blood cells. It's full of these defense cells. And the white blood cells are able to move in and out of our blood. But when they need to get to an area fast, they can use the lymph system to travel quickly. Think of them like freeway systems. So, your blood flow is kind of like a freeway. The lymph system's right beside it. Think of that as kind of like the carpool lane. Okay, and if you've ever been in like LA during a traffic rush, that carpool lane is still flowing. That's kind of how this lymph system works. It's still flowing. It allows our first responders to quickly move from one area to another. It's like a faster freeway that they can use. So being able to move through the lymph system is very important. This is also where we're going to have a lot of our antibodies traveling. So the antibodies can also travel through the lymph system, which we'll talk about in our next chapter. Now, the lymph system then is going to be composed of these little freeway passages, and along the way, they're going to have special nodules that we call lymph nodes. Think of these as kind of like a rest stop or like a little train station. The idea with a lymph node then is it's an area where we can kind of gather any bacteria that the white blood cells have encountered or any viruses, and they kind of move them to this general location. So think of it kind of like I said, like a police station or a rest stop. It's a general location where we can have a gathering. Now, if I have a white blood cell, I can grab a microbe, bring it with me to the lymph node, and then when I get to the lymph node, I have other white blood cells that can help me annihilate it. Okay? So I'll have a whole bunch of white blood cells sitting in this lymph node region waiting to help. So it makes it a faster and easier way to get rid of a whole large number of microbes. The problem with the lymph nodes, though, is that we can have bacteria and viruses accumulate in such a high number that they can swell. This is why when we get sick, if we get a strep throat, sore throat, anything like that, we can feel the lymph nodes and they'll feel swollen. Most of our lymph nodes are found under our throat, in our armpits, in the groin region. This is why, for example, during the Black Plague, they were talking about people having buboes. Buboes were black-colored lymph nodes. And so what was happening was the bacterium that causes the Black Plague, does anybody remember which one that one was? Excellent. Yersinia pestis. Yersinia pestis. Yersinia pestis was a bacterium. Our white blood cells would grab it, move it into the lymph nodes. It would start causing destruction in the lymph node tissue. So we would see a blackening. And then it would cause a swelling. Those are referred to as the buboes, the Black Plague. That's why it was called Black Plague. It was black because we would see a darkening color on the buboes, on the lymph nodes. One of the things that they did as a treatment was they popped the lymph nodes. Was that a good idea? No, that usually resulted in instant death. Because what that did is it released all those bacteria that they had just locked up. Yeah, Jasmine.
Is it common to have lymph node swelling after you get vaccinated?
Yes, it is very common to have lymph nodes swell after vaccination.
Yeah.
Why? When I have a vaccination, what am I producing a whole bunch of? Antibodies and defense system responses. And so what's happening is those antibodies are reacting and they're moving to the lymph node going, look, I just got exposed to this antigen. It's like they're sharing the most wanted poster. Does everybody see it? And then other antibodies are like, yeah, and they're building up in that area. And so it can cause a little bit of swelling. So a lot of times after vaccination, you can get swollen under the arms, especially. A lot of times it's near the armpits or near the neck. Yeah.
So your tonsils serve as like a first defense, right?
Yes.
People who get them removed, does that mean that they're somewhat immunocompromised?
Great question. So we used to think tonsils didn't do anything.
Right.
And now we've learned that tonsils do serve as kind of like a source of where we can kind of store bacteria as we're fighting them. So they can get inflamed during infections. So people who have had their tonsils removed, they're still okay. They're not immunocompromised because we have all these other defense systems. The tonsils helped, but it's like removing one lymph node as opposed to removing all of them. So the other ones kind of up their game to help compensate. But if you've noticed, used to, if anybody got a sore throat, it was like, oh, remove the tonsils. And nowadays we don't do that. Nowadays we're much more careful. It's like, we kind of do it as a last resort. Because we've learned that it is better if they can keep their tonsils as best as possible. But unfortunately, sometimes they just keep swelling and getting too big and they have to get out. But they're not immunocompromised because we do have so many other things that help. Same is true, for example, with the spleen. The spleen is where a lot of our lymph is going to pass through. And the spleen is kind of used as another area of gathering for white blood cells and another area of learning. We found that when the spleen is removed in people that are under 18, that it affects their immune system more than in people that are over 18. And let's think about why. If I'm under 18, is my immune system still learning a lot? Yeah, we're still getting exposed to a lot of new microbes. We're still getting exposed to a lot of new viruses. So the spleen is kind of like a community center where antibodies, white blood cells can come together and learn from one another. So share those most wanted posters, share information. If I remove the spleen from a child, I've removed their community center. So there's less learning. So they do tend to have immune systems that don't act as fast when they're adults, which is one of the reasons why they try to not remove spleens in people that are under 18. If you're over 18, we find that that's usually kind of a golden era because your spleen has already seen it all by that age. You've already been exposed to pretty much everything. So it doesn't seem to impact you as much. And remember, we talked about what our average lifespan should be, right? So it kind of makes sense that 18, that's kind of that middle range of life for most people back in the old days, right? So that was kind of like midlife. Can you imagine that? At 18, you're like middle age. I mean, come on. But that was the idea, is that if they're younger than that, they're not going to have as much of a learning process. So, hello. Okay, let me get back in here. Just a second, down Zoom. Did I have anybody in there? I don't think I had anybody in there. All right, we'll just do that. Okay, so this is kind of what our lymph system looks like. So here we have right here our deoxygenated blood. Here we have our oxygenated blood. So these are like little freeway systems. And then right in the middle, we have the lymph system. And again, think of this as like a carpool lane. If I get a cut, my white blood cells can hop in that lymph system and zoom to where they need to go. And then they can quickly move across into the bloodstream if needed. So they're constantly moving back and forth. Think of it as kind of like a policeman on patrol. They can patrol the blood system and then pop back into the lymph system if they need to move somewhere quickly. Their big goal is to try to isolate microbes. So if they come across something, they're going to try to pull it into the lymph node or pull it into the lymph system to get it out of the regular blood flow. Does that make sense? Think about where your blood flow is going. Is it going to the heart? Is it going to the brain? Do we want to protect those regions? Yeah, so if we can put something that's dangerous in a lymph node and isolate it, we have a much better survival chance than letting it sit in the blood and go to the brain or the heart. Okay, so we want to use this lymph system a lot. Here's a picture of a typical lymph system then throughout the body. And you can see that there's the little lymph nodes that are under the armpits, that are up at the top under the neck, that are down here in the groin region. So these again are going to be accumulation points. And the idea is that there's a whole bunch of white blood cells waiting in that area to help annihilate whatever is causing an infection. Now, can things go wrong with the lymph system? The answer, sadly, is yes. Here's a picture of when things go wrong with the lymph system. This is a little worm called Wucheria bancroftii. And Wucheria is found naturally in Africa. It's associated with water. You usually get it through drinking contaminated water. And this little worm, you'll drink it as an egg, so you don't even see it. It's microscopic. And the eggs will move into the body and they will move into the lymph system. They'll travel in the lymph system and they manage to fool our immune system so that the immune system doesn't trigger. We don't know why, but our white blood cells don't react. The little worm hatches out and it ends up starting to grow and it gets stuck in the lymph system. It's basically like a car crash on the carpool lane, okay? So now I have this worm and the lymph that normally would flow starts to back up. And what we see is that they usually start to back up in the lower extremities, so the lower legs. So again, it's like a car crash. I can't get around this worm, so the lymph just kind of pools. It doesn't circulate. Now, as you can imagine, this is very uncomfortable. This is like, major edema of the tissue. And can it be treated? And the answer is yes, we can treat it. But a lot of people don't seek treatment because usually if you get treated and it clears, you just get reinfected again. So it's something where a lot of times in some areas, they just don't really even care at this point. They're just kind of like, well, what's the point of treating it because I'm just going to get it again. Also, for their culture, this is very normal. If they see an adult with two thin legs, they know that you're an outsider. They expect you to have, at least some swelling in your legs by the time you're an adult because that's normal in regions that have Wucheria. So we see this and we think it's abnormal. They see it and they think it's normal because that's just part of their region that they live in. So again, we can treat it, but it's hard to get rid of. Okay, compare the structures and functions of monocytes and neutrophils. Which are not phagocytotic in the bloodstream? Monocytes or neutrophils? Monocytes, excellent. Which become macrophages and move into the deep tissue? Monocytes or neutrophils?
Monocytes.
Which ones?
Monocytes.
Monocytes, excellent. Which is the first to an infection?
Neutrophils.
Neutrophils, good. They're our first responders. So it's monos, and then neutros. What is the function of the lymph nodes? What do the lymph nodes do?
It's not.
Do you guys not have this slide?
So bacteria.
Oh no, take a picture of it. Yeah, take a picture of it and then put in your notes and say
What is that?
AAB. Yep. And then what is the function of the lymph nodes? Who can tell me?
A gathering site?
Excellent, a gathering site and a learning center, right? Excellent. Sorry, I didn't think you guys had that. Sorry. So take a picture of it. Do take a picture of it. If you don't have your phone with you, ask your lab partner if they can send it to you. And then put a note in your notes that the answers are A, B, and then gathering site. Sorry about that. Okay. Yes, gathering site and learning center. Excellent. Okay, let's talk about our second line of defense a little bit more. Let's go into a little bit more information about those white blood cells. So, as we mentioned, a lot of our white blood cells are able to do phagocytosis. And phago in Greek means to eat, and site means cell. So these are our cell eaters, kind of like the Death Eaters in Harry Potter, right? So they're the cell eaters. So they're able to do phagocytosis, which means they can ingest any invader and destroy it. And the idea behind this is that they're going to try to get rid of as many of these invaders as possible. Now, the way this works then is if an invader is found.
Could you go back to that one?
Yeah, pretty quick. Sorry. If an invader is found, these cells are going to go up to the organism that's trying to invade. They're going to wrap around it and pull the organism inside of them. And then once they have eaten the organism, they're going to break it down and discard it. So here's a picture of that process. So here we have a little diplococci. Our white blood cell is approaching. It grabs onto the diplococci. I love this picture. Doesn't it look like little eyeballs? It looks so cute, right? And doesn't he look like Kirby? How many people remember Kirby from the Nintendo days? Yeah, this is like Kirby. He's so cute. Anyway, okay. Yeah, so Kirby. Kind of looks like that, but he's pulled the diplococci inside. Now notice it hasn't destroyed the diplococci yet. So they will engulf the microbe or the virus. They'll pull it inside of them to get it out of our bloodstream, to get it out of the lymph system. It's now inside of them. And then their job is they're going to release chemicals inside their cells to kill those organisms. But that's the next step. So the first step is to just engulf them and get them out of the way. So during an infection, then the neutrophils and the monocytes are going to move to the infected area. They are going to quickly grab every microbe they come across. So they're highly phagocytotic. They're grabbing microbes right and left, pulling them inside. And then what's going to happen is the macrophages are going to come in and they're going to help clean up any cell debris left behind. So let's go through this process. I have a bacterium. Maybe I cut my hand and I have bacteria coming into that cut. My first responders are the neutrophils. They're on site, they're grabbing any bacteria that have entered that wound. Now, sometimes, unfortunately, when they pull in these microbes inside of them, it's going to result in their own death. So if they have microbes inside of them, they're going to release chemicals. Remember the lysozymes? Yeah, they have little suicide sacs that they release that's going to take out the microbe. But is that going to kill them too? Yeah, it can kill them too. So they basically sacrifice themselves. So now what happens is we get debris left behind from the neutrophils. Their leftover cell debris and the microbes leftover cell debris. Now we have the macrophages that come in as the second line, and their job is they're the cleanup group. They like pick up all the cell debris, get rid of it, clear it out. They're cleaning up the car accident. You see people on the roads and there's a car accident, the second responders are the ones clearing it up. That's what these guys do. So our first responders, we know they're active because we'll see a lot of cell debris. We visually see that as pus. You guys ever seen pus? Or if you've had pimples that have had pus in them, right? The idea then is that white pus that you're seeing, those are your neutrophils that have died in your honor. Isn't that special? I mean, like, you look at pus a little differently, right? I mean, we look at it and we're like, ew, and our poor neutrophils are like, I died for you. And you said, ew? Like, whatever, come on. But that's what they are. They're basically cells that have died for you. And so what we're seeing is the debris that's left over. Now what'll happen is over a few days that pus goes away because the macrophages come in and they clean it up. They come in with their little vacuum cleaners and they're like cleaning it all up. Get rid of it. Maybe not a vacuum cleaner, but you get the idea. Okay, so we have several ways for phagocytosis to work. The first is that our cells will follow chemical patterns or chemical traces of microbes. So they are attracted to cells that are different. So because of that, they can tell where a microbe is and they can kind of follow it to grab it. Also, when they destroy these microbes, they leave behind more of these chemical traces. So a neutrophil grabs it, dies with these microbes, and now I release a whole bunch of these chemicals that calls in more neutrophils, that causes in more macrophages. So that's chemotaxis. When they come across a microbe, they're going to adhere to it, they're going to stick to it, they're going to ingest it, they're going to pull it inside. Now the microbe's immediately going to be moved to a special compartment called a phagosome. The phagosome is a little organelle that we find specifically in white blood cells that can fuse with the lysozyme. The idea behind this is if I have my microbe in the phagosome and my lysozyme is nearby and they fuse together, then my white blood cell can survive. The lysozyme's only fused with the phagosome. Everybody with me on that? But if I have a white blood cell that has gathered a whole bunch of microbes, let's say it has about 20 or 30 microbes that it's pulled inside of it, rather than do that process, it's just going to burst its lysozyme and kill out as many as it can. The idea is kind of like the difference between taking one prisoner or throwing a grenade. And that's basically what they're doing. They release the lysozyme and they get a grenade if they have enough microbes inside of them. Again, sacrificing for the greater good, which is, it's like a battlefield out there. There's actually, I'll have to find the video. There's a video that's on YouTube that was created by a doctor that talks about your defense system. And he has these cells as little characters. And it's really cute because it walks you through all the different white blood cells. And it's like a little anime one. So they all have like different personalities. It's pretty cute. I'll have to see if I can find the link and give it to you guys. And then we get digestion. So the ones that are able to have a small number and survive, they'll destroy the microbe. But here's what's interesting. They will hold in the material that's left over on their cell surface as kind of like an instigator or a target to show other white blood cells what to look for. So it's kind of like, hunting, and you have people that have trophies of hunting. That's kind of what they're doing. They're holding out a little trophy like, look what this is. If you see this, attack it. And then other white blood cells see it, and then they start attacking. Isn't that fascinating that our cells can do all this? It's amazing how they work. So here's a picture of how that process works when all is going well. I pull in a microbe. It goes directly into my phagosome. The phagosome is a little protective organelle. The lysosome comes and fuses with it and destroys just that little microbe. Now what's going to happen is I will excrete the chemicals that that microbe had, kind of like a little trophy to show other cells what to look for. So they know what the chemotaxis line is, and they know what to try to find. Again, this works great if I'm only pulling in a small number of microbes. If I pull in a whole bunch, I'm going to skip this process and just burst open the lysozyme and take out as many as I can. Okay? So this is great, and this works really, really well in most cases. Unfortunately, we do have a few microbes out there who have learned ways to evade the system. So, for example, we have organisms that can produce capsules. We talked about capsules before that make them bigger. This makes it harder for the white blood cells to digest them or to try to eat them. So it's a way for them to evade and survive. So organisms like Streptococcus pyogenes, Streptococcus pneumoniae, they have these big capsules that are like shields around them, and our white blood cells can't figure out how to eat them. They're too big. So if I can't get eaten, I can't get destroyed. Some cells will get phagocytosed, meaning they'll go inside the white blood cell, but then they attack the white blood cell and turn around and kill it. So organisms that do this are things like staph and listeria. So they're like, oh, you want to eat me? Okay, go ahead and eat me. I'm just a harmless cell. And they get pulled inside, and then they're like, ah, and then they attack the cell and kill the cell. Isn't that evil? Yeah, so I mean, these are like evil bacteria. So listeria and staph do this. This is why listeria is dangerous for developing fetuses. Remember we talked about it can cross the placental barrier. If I'm a developing fetus and I have new tissue that's forming and I have listeria in it, guess what listeria is doing? Popping the cells. When I'm a fetus, I don't have that many cells to begin with. And listeria is actively destroying my cells. It can cause birth defects. It can cause death because it's destroying the cell. Some organisms have learned to escape the phagosome. So they get engulfed, they go into the cell, they get inside the phagosome, but then they quickly release chemicals that prevent this phagosome-lysome fusion. They release chemicals that make it so the lysosome can't attach to the phagosome. They basically put a barrier up. Organisms that do this are Shigella, Rickettsia, HIV, and tuberculosis. So what this means then is that they're inside the white blood cells, but the white blood cells can't kill them. So what happens is I get increased inflammation, I get increased irritation, I get increased white blood cells trying to get rid of the infection, but the infections don't clear on their own. Does that sound about right for these organisms? Yeah, sounds about right. Okay, and then some can survive in the phagosome even when the lysosome comes in. So these guys are just so hardcore. They have these cell walls that protect them. They can survive. One of these is Coxiella that causes Q fever. Thankfully, Q fever is very rare. Remember we talked about it when we talked about prokaryotes, and I told you it was very rare, but we always keep an eye on it. And the reason for that is because it is one that is completely immune to our defense systems. So that we have to give doses of antibiotic to get rid of it because our white blood cells on their own cannot. Okay, let's talk a little bit about inflammation. So we tend to think of inflammation as bad. And we need to kind of change the way of our thinking. Because inflammation isn't bad, it's just a response. It's your body trying to protect you. So if you get an injury or an infection, one of the processes that your body will do is it will have inflammation occur. The idea is it's trying to isolate the pathogens in a region that's inflamed, and the inflammation is going to allow more white blood cells to come into that area. Now, if we look at typical inflammation, we'll see that it's characterized with redness, with swelling, and if I touch the inflamed area, it also heats. So those are the typical characteristics of inflammation. So let's look and see why. The first one, redness. The reason we see redness associated with inflammation is because our blood vessels start to dilate. So our blood vessels are going to expand a little to allow more blood flow to the region, hopefully to allow more white blood cells to get in the region. So that's why we start to see redness. We see swelling because plasma from the blood and white blood cells start to accumulate in the damaged tissue. So it's kind of like if you've ever seen an accident, like a car accident, you'll see first of all a police car show up and then a fire truck show up and then another police car show up, and before you know it, there's a party, right? There's all these people around. That's what's happening here. We're getting swelling because all these cells are showing up to the area, okay? We start to have pain. Why do we have pain? Well, we have swelling. We have all these cells trying to accumulate in a small area. That's pushing against our tissues. It's pushing on pressure sensors now, and it's pushing on pain receptors. So it feels uncomfortable. And then we start to get heat. We feel hot because of that blood flow increase. That dilation is going to cause that heat to increase because we're getting more blood flow to that region. Now, there's two different types of inflammation. We can have acute inflammation, which means it's short and quick. So let's say I have a knife cut. I always cut myself when I'm chopping veggies. You know, no matter how careful I am, there's always like a wound involved. So you might get acute inflammation, so a little bit of swelling at that site. Maybe if you have a broken bone, you can get acute inflammation at that site, you know, where there's damage. We also have some people that have chronic inflammation. This is much longer lasting. It tends to be less intense. So I don't really always see redness anymore. I don't always see, you know, like pain associated. Instead, what I just see is a visible sign of swelling or increased tissue size. This could be something like maybe a bone didn't heal properly, so the white blood cells still kind of stay in that region. So there's always a little swelling or puffiness there. Another example would be arthritis. Arthritis is where the joints are inflamed, so there's always a little swelling there. It's not from any injury, it's just your body's reacting to the discomfort there. So with this, the stages of inflammation then are all to try to protect us. So here, if we see a cut, we see that we have bacteria sneaking in. The first thing we're going to do is we're going to increase blood flow. When we increase blood flow, that's going to bring with it all of these white blood cells who are going to start trying to defend the wound site. So these would be our neutrophils, our first responders, coming in, trying to grab any organisms that they can and get rid of them. As they move to the area, that's going to cause edema or swelling because their numbers are increasing near that injury site. That also is going to have redness and heat because of the fact that we're getting more blood flow to that region. Now, after the neutrophils show up, we also get additional flow of our macrophages and our other white blood cells that are going to try to make sure that they work together to destroy any bacteria that have come in. So neutrophils are grabbing, this is a neutrophil here, grabbing bacteria. If they grab a whole bunch, they release their lysozyme and take out themselves and the bacteria. The macrophages come in and they'll remove these dead cells as well as the dead bacteria. So they're going to help annihilate everything together. What we should see then is over time, we'll have the creation of pus in the area. That's just where we have debris of cells. And then we should start to see inflammation decrease and tissue repair occur at the site of the wound. Now, the amount of time that this takes really depends on how quick of an immune response you have and how healthy your tissues are. For example, we know that diabetics tend to heal slower. Why? Because their cells are already having issues with absorption of sugar, they're having issues with absorption of nutrients, so they tend to have slower responses with their immune systems. So because of that, if a diabetic gets a wound, it's going to take almost twice as long to heal as somebody who's not diabetic. This is why we worry about people who are diabetic with wounds on their extremities. We always have to keep an eye on them. Okay, should we do one more? Should we do fever? Or you guys want to stop here and do fever next time? Are your brains full? Should we stop? Keep going. Keep going? You gotta keep going? Okay, you guys can do 9 more slides? All right, okay, 9 more slides. Okay. All right, the next response we're going to have is going to be a way that we can do fever. Now, fevers also have kind of a bad rap when it comes to things that are like negatively viewed. A fever is simply an abnormally high body temperature. So it's where our body starts to go higher. Now, let's think about why our body would do that. Do microbes have a preferred temperature setting? Remember, we've talked about that, right? We know microbes grow best at certain temperatures. It turns out most of our pathogens grow best at about 37 Celsius. That's what we're usually set at. That's body temperature. So if my hypothalamus goes higher, my hypothalamus, think of it as like the thermostat in your brain. If it sets itself higher, what's going to happen is body temperature will go up, and the idea is that we're trying to cook out or get rid of the microbes. So if your white blood cells are feeling overwhelmed, if there's too many bacteria present, they're going to send a signal to the brain saying, we need some help. The hypothalamus is like the thermostat in your brain. Think of it like the thermostat in your house. When you get cold, you turn the heat up. That's what the hypothalamus does. It goes, okay, we're going to turn the heat up and try to cook these guys out. It's going to turn the heat up a few degrees. So we might go from a normal setting of 37 to a setting of 40. Now that means that our body temperature is going to go 3 degrees higher. So if I'm at 98, I'm now at a 101 or a 102. Is that okay? The answer is yes. If my fever goes up a few degrees, I really want to try to avoid taking Tylenol. If I'm at like 100, 101, 102, that's okay. People kind of panic. They're like, oh my gosh, I have a fever. I have like a 99. I got to take a whole bunch of Tylenol or Advil and get this down. When you do that, you are absolutely destroying your immune response. Let your immune system do what it needs to do. What it's doing is it's trying to cook out the microbes. And so having a low fever is actually healthy. We are killing a whole bunch of microbes during that time. Now, if you get past that 103, 104, that's where you should start medicating. I wouldn't go past like 103.5. Like that's starting to get too high. That you want to bring your fever back down. But if you're at a low fever, let it ride. It's okay. Your body's trying to help you. Now, what happens when we get a fever? Well, the first sign of a fever is chill. Doesn't that seem kind of the opposite? Fever's getting hotter, right? Well, the reason we get a chill is because our body has reset its temperature. The hypothalamus is going hotter. So now our body is like, oh, I'm at 98.6, which is the same as 37 Celsius. I'm at this temperature and it's not good enough. So now I feel cold because my body's reset higher. So now I'm going to try to get warmer, right? So I have the shivers. That's going to help me heat me up. I might cover up with a blanket. I might try to get warmer. So I'm increasing temperature. Where I run into problems is that sometimes we end up with a situation where we get a positive feedback loop. Now remember, in most of our biology, we get negative feedbacks. I increase the temperature, I kill off the microbes, I turn the temperature back down. A positive feedback loop is what happens in my house. I turn the temperature up to 68, my kids come behind me and go 70, my next kid comes around and goes 75, my next kid comes around and goes 76. I only have 2, but still, it feels like I have 50. They keep turning the heat up, right? And so what happens, it's like all of a sudden we're all sweating in this house. It's like, why is it so hot? Because they kept turning it up. That's a positive feedback. We kept going the same direction. So a positive feedback loop can happen when the hypothalamus keeps getting signals from the body that there's too many bacteria or too many viruses, and it goes, okay, we'll turn it up some more. And we keep turning it up. Now we're at a temperature of like a 105. Now we're at a temperature of 106. What are we running the risk of with our proteins? Yes, denaturing. We're getting too hot, which is why we need to take Tylenol or Advil to back it down. Yeah, it's time.
What's the temperature where your brain starts to get like affected?
That's usually around 109. It can actually go up pretty high. 109 to around 112 is where we start to see issues. So we can actually get pretty darn hot. This, though, is interesting because in kids, we see a lot of times where they'll have febrile seizures. And a febrile seizure is terrifying to witness. It is actually harmless. What it is it's like rebooting your computer. The brain realized it was getting too hot, and so it shuts down. That's the seizure. You see that you start twitching, or your eyes go up in their head. It's scary to watch. What's happening is the brain is turning the hypothalamus off and turning it back on, like a reboot. And that lowers the temperature. So that's what a febrile seizure is. It's where we got too hot, we have a febrile seizure, and we reset. Yeah.
What if the child has it very commonly happen?
Exactly. So sometimes we have a hypothalamus that's very sensitive. So as soon as it starts heating up, what you'll want to do is you'll want to monitor that child and find out what their threshold is. Usually for most kids, it's around 106. But you're right, some are super sensitive. When they hit around 104, they can go into a seizure. So you don't want to let that kid get past 103.
It's different because my son seized at 100. He wasn't even.
So again, super sensitive, and it depends on microbial count load too.
Yeah.
And it's everybody will have a different setting. So that's what's in. You have to kind of learn what's right for your kid. So, like, for my family, I'm like the most unsympathetic mom. My kids are like, I have a fever. I'm like, you're living, you're fine. Good for you. Good job. Your immune system's kicking in. And they're, I'll check them and they're like 101. They're like, I need Tylenol. I'm like, you're fine. Wait till it's a 103 and then talk to me, So, because I'm like the mean mom. I'm like, I'm checking them. But I'm like, no, we're letting your immunity work, I'm also the mom that, like, one of the things that they used to tell you is give your kids Tylenol or Advil before a vaccine. Is that a good idea? No. It tends to lower your vaccinations. So like when you get your COVID vaccine, try to not take Tylenol or Advil for several hours after your vaccine. Hopefully try to not take it for a full day if you can. If your arm's too sore, wait at least 12 hours. And then the idea is that when you do take it, it stops your antibody production briefly. We don't want that. That's why sometimes people will get vaccinations and then they'll say, oh, it didn't work. I still got sick. What's that?
Why do they, on the CDC, I've seen it, let's say, take time off your meeting the same day.
Exactly. So it's usually several hours after. But keep in mind, CDC is always learning. We weren't supposed to wear masks, and now we are. We're learning it affects antibody counts. Okay. So it's something that pediatricians used to tell parents about 2 hours before you come in, give them Advil or ibuprofen. Now they don't do that. And they shouldn't, because it stops your antibody. It doesn't stop it, but it lowers it. So fevers are not bad. Inflammation's not bad. I'm out of time. So we'll pick up on this slide next time. We'll have 7 more slides, and then we'll go from there.